Thank you so much for the introduction, and thank you also for the invite. Very honored to participate in a great initiative like this one, and to be able to share some of the work that we've been doing recently in the Tavazoy lab, studying the diversity that we see in microglia in late-stage Alzheimer's disease. And so this project really began with our lab's very long-standing interest in apolipoprotein, or APOE, which is a very well-known secreted glycoprotein, highly pleiotropic, involved in a really diverse range of biological processes. For people that are familiar with apolipoprotein E or APOE, you'll remember that it comes in three major variants in the human population: APOE2, APOE3, and APOE4. E3 is by far the most common. It's about 75% of all the alleles in the human population. And then the remaining 30 are roughly split evenly between APOE2 and APOE4. And these variants only differ at 2 amino acid residues. They're really quite similar. But in spite of that, they have massive implications for downstream diseases. And so our lab, and in particular, one postdoctoral student in our lab who has since gone on to establish his own group at the Charité in Berlin, Ben Ostendorf, had this really beautiful work, seminal work in 2020 that came out in Nature Medicine, where he showed that if you just took mice, black 6 mice, no additional mutations, and just replaced the murine APOE locus with human APOE2, 3, or 4, then he found that these APOE2-bearing mice had much more rapid melanoma outgrowth when you just did subcutaneous implantation of melanoma, and APOE4 mice had much slower melanoma outgrowth. And he found that all you needed to do to recapitulate this in the absence of this sort of whole body mutation was to just do a bone marrow transplantation, and that was sufficient to sort of phenocopy this effect. So clearly, APOE is regulating anti-tumor immunity in some capacity. And he then did follow-up work 2 years later at the peak of the pandemic and showed that the same was actually true for SARS-CoV-2 infection. And so, of course, elderly mice were more likely to die of COVID infection. He found that males were also more likely than females to die of COVID infection, which epidemiological studies have also carried out. But what he found, which was really fascinating, is that E4 mice actually succumb to disease more rapidly than E3, and E2 also succumb more rapidly to disease than E3. And it's very well known, of course, that in the context of Alzheimer's disease, APOE4 is among the largest risk factors for Alzheimer's disease progression, in addition to age, right? So as you get older, you're more likely to get Alzheimer's, but if you carry 2 alleles of the APOE4 allele, if you carry 2 copies of it, you're much more likely to get Alzheimer's. It's something like a 60-fold increase in your likelihood compared to if you have E2, for instance, which is protective in this context. And so coming into this project, when I joined the lab, we had this sort of central burning question, which was, if age and APOE are the 2 major risk factors that define Alzheimer's, how do they jointly modulate disease progression? And in particular, given our interest as a lab in APOE as an immunomodulatory molecule, how did these 2 factors impact microglia, which are the main immune population of the central nervous system, and in particular, key mediators of Alzheimer's pathology? And so we approached this question with a very straightforward opening approach. What we did is we developed these mouse models, starting with the same model that Wei used in his paper that he described just before me very beautifully, this 5x FAD model, also called 5xTG, which is a common model of Alzheimer's disease. And we crossed them to mice bearing APOE2, APOE3, or APOE4 knocked into the murine APOE locus. And we aged these mice to 10 weeks of age, 20 weeks of age, or 96 weeks of age. Now, I should emphasize here that this 5x FAD model is a very aggressive model of Alzheimer's disease. By 20 or at least 25 weeks or so, you can already very reliably observe robust emergence of amyloid beta in the brain. You see behavioral defects. And so the reason why we chose these time points is that at 10 weeks, you have this pre-pathological state. At 20 weeks, you already have onset of pathology. And 96 weeks is a true, really terminal state pathological disease. Most mice die well before 96 weeks, so we had to start with enormous cohorts of mice to have enough at 96 to actually do the sequencing. And then we harvested the hippocampus and the cortex from the brains of these mice at these time points, because this is the particular regions of their brains that are most involved in Alzheimer's pathology, and used flow sorting to isolate CD45 positive cells, which are all the immune populations you'll get from the brain. As Wei described, you'll mostly get microglia from this, but you'll also get some other immune populations like B and T cells, neutrophils, macrophages, some that might come in from the meninges and the dura, and these lymphatics, as Johnny Kipnis described in his work about a decade ago, some that might have come in from other routes. And then we performed single-cell RNA sequencing to examine the heterogeneity that we see in these APOE variant and age-dependent single-cell immune systems. And what we found, zooming into the microglia in particular, because these are about 80–85% of the cells that we get, is that there was a really striking degree of heterogeneity. And here I'm showing you all of the microglia that we got across all of our APOE variants and all of our time points. And in general, there are 4 large superclusters that we identified. The top left, you can see this homeostatic group. These are the resting microglia that enrich particularly in 10-week-old mice, as you would expect. you have this inflammatory supercluster that includes more acutely inflammatory populations and also more chronically inflammatory populations like the disease or damage-associated microglia that have been described by Ito and Mead and are very well established in mouse models of Alzheimer's. We had a small population of stressed microglia that expressed heat shock proteins, but the one that really caught our attention was this group that you can see at the bottom right that we labeled TIM, or terminally inflammatory microglia. And I'll come back to why we called them that in a bit. But what was really exciting to us about this population is that it was almost exclusive to mice at the 96-week time point. You can really appreciate this here. At 10 weeks, they were almost all this homeostatic state in the top left. At 20 weeks, we started to see occupancy in this inflammatory state. At 96, it's almost exclusively these TIM. And right away, we were curious, what makes these TIM unique compared to the other microglia that we identify across this entire atlas? And so we did the natural thing. What expression do they have that's distinct? And we found that they were depleted for homeostatic markers of microglial state, like P2RY12, but they were also massively enriched for a combination of stress markers like FOS and JUN and other AP1 factors, as well as inflammatory markers like S100A8, S100A9. So right away, we kind of had this very interesting idea that these microglia seemed to be like a stress inflammatory state. They had this inflammatory phenotype, but they were also taking on this other weird phenotype that hadn't been described in the past. And what was really exciting to us is that when we were able to do directed fate mapping using cell rank, which is a combination of RNA velocity as well as a number of other factors, to model the coalescence state of the system. So if you start with the microglia that you would expect to be a homeostatic cell, and you ask it in the system, you run the tape all the way to its conclusion, what is the terminal state for cells in the system? What we found is that very reliably, these cells would start off homeostatic, progress into this inflammatory state, and then end up in this TIM cluster that we identified at the bottom. And so these microglia were very clearly a terminal state and an inflammatory state. So we called them terminal inflammatory microglia. And this was sort of the key onset for all of the work that we did in the rest of this manuscript. Now, what was really exciting to us, in addition to what I've already described about these TIM, is that they had very differential enrichment across APOE genotypes. And so, for instance, if we looked in APOE4, there were much more of these TIM than we saw in APOE3 or in APOE2. And of course, thinking about the fact that APOE4 is also a risk factor for Alzheimer's, this start to give us a little bit of insight as to what might be driving the differential risk that people have with these different genotypes in Alzheimer's. The other thing that we found to be particularly interesting is that TIM are not all made equal. There were two very clear distinct subpopulations of TIM that we identified in our single cell atlas. Some of these were effector low TIM, and these TIM expressed even higher levels of these stress markers. They had low expression of effector molecules in inflammatory states, whereas effector high TIM still retained functionality, or at least from their transcriptional state, it appeared that they retained functionality. And what we did to really get at this question a bit more of how are these distributed across genotype is we took another cohort of 15 mice from E2, E3, and E4, and we did bulk RNA sequencing on microglia from their brains and used our single cell atlas to decompose, to predict what is the fraction of each of these different populations that are represented in our bulk sequencing. And from this, we were able to find that as our atlas predicted, APOE4 massively enriches for these effector low TIM, whereas APOE2 enriches for these effector high TIM. And so the story that begins to emerge here is that in APOE4, you get a lot of these very, very sort of low ability, low function effector low TIMs, whereas in APOE2, you get less TIM overall, and the TIM that you do get are this effector high state that still maintains functionality. Now, we did the natural next couple of steps, which is to try to characterize the cell population in a little bit more detail. One of the things that we did was to model which transcription factors were defining these transcriptional signatures and states, so we can compare TIM to non-TIM microglia in the data set. And what we found reliably is that TIM were very strongly enriched for AP1 factors. You can see across the board here, KLF4, KLF2, FOS, JUN, JUN-D, all these different factors that participate in AP1 signaling were very strongly enriched in TIM. We could also ask, much like Wei did, what are the ligands and receptors that are being used to define the interactome of the system? What we found is that TIM massively enriched a massive number of ligands and receptor interactions, IL-11, DHEA, 2AG, that are responsible for tamping down the immune response, for reducing inflammation and activation. And in addition to that, what we found that was very striking is that TIM actually are much less promiscuous in their interactions in general than other microglia are in the system. You can see that in the circus plot here to the right. The size of the bar here represents the total number of interactions that are made by a cluster. And you can see that the size for these TIM populations is much smaller than the sizes for some of these other homeostatic-like or inflammatory microglia in particular. And so these TIM appear to be not just this sort of stressed population, they also fade into the background. They don't make as many interactions as other microglia, healthier, happier microglia would. And the last thing that we did, well, one of the other things that we did, there's much more in the paper that I won't get to today, is we tried to infer the metabolic state of these populations by solving the flux balance analysis for all metabolic reactions in the cell, weighted by their expression of different metabolic enzymes. And what we showed is that TIM massively enrich for amino acid processing pathways, but they deplete for a lot of bioenergetic pathways. For instance, glycolysis, pentose phosphate, galactose, lots of different sugar processing. And this stuck out to me because I'm a computational biologist, but I'm also an immunologist by trade. And as soon as I saw, you have a stressed cell state that has massively depleted its interactions, it overexpresses these inhibitory ligands and receptors, and it reduces its utilization of glycolysis, that begins to sound a lot like an exhausted T cell like you would see in the tumor microenvironment. And we're a cancer biology lab, and so I'm always primed to be thinking about the tumor microenvironment. And so to me, right away, the model that I began to develop in my mind was that these TIM kind of represent an exhausted-like microglial population in Alzheimer's. And that would make a lot of sense. You can kind of imagine that these microglia have been marinating in the milieu of the Alzheimer's brain for 96 weeks at this point. They've sort of been inundated with this chronic inflammatory stimulus, and over the course of this period of time, they begin to take on this more exhausted state where they're no longer able to respond to the inflammatory stimuli in a constructive way, much like an exhausted T cell in the tumor. But we really, at this stage, didn't have a good sense of what are the specific molecular drivers that produce TIM. And the reason for this is we had this 20-week time point where you see no TIM, and the 96-week time point where you basically see only TIM. We didn't really have a sense of what happens in between, what leads to this emergence TIM. And so what we decided to do was to leverage a technology called multiome sequencing, which allows us to get both RNA and attack or chromatin accessibility information simultaneously at single cell resolution. And this really gives you an unparalleled level of information that allows you to model the gene regulation at a single cell resolution. So we did this in some mice that were bearing APOE4 in the Alzheimer's model at 60 weeks of age. Rationale being that E4 expresses more TIM, and so the 60-week time point would be the most likely to give us TIM as compared to E2 or E3, which might not have that many TIM. We were very relieved to find that at the 60-week time point, we get this very nice cluster of TIM coming off the main microglial population here. And if you look at the emergence of TIM and the frequency of TIM over these different time points, at 10-week to 20-week, it's basically nothing. You can see it here zoomed in. It only goes up to about 0.3% of all microglia in the data set, whereas in that 1-year data set, it was closer to 7%. In E3 at 2 years, it's about 42. E4 at 2 years, it's closer to 70. And so here we have this very good sense of the natural progression over time. As the mouse gets older, there are more and more TIM present in its brain. And so we now have this RNA and attack information. And we leveraged a lot of different tools to try to identify what are the specific transcription factors and chromatin regulation that are driving the TIM state. First thing that I did was to use latent Dirichlet allocation to do topic modeling and identify RNA and attack features that are co-regulated and define a particular microglial state. And so, for instance, topic 2 here on the left is particularly enriched in TIM. And we found that that was marked by expression of RUNX factors. So RUNX 1, 2, and 3, as well as BGB, were all sort of defining this TIM topic that was co-regulated. Whereas topic 3, which was enriched in DAM, was instead enriched for CBP transcription factors, as well as some AP1 signaling. But the deeper question that we wanted to ask is, okay, well, what are the enhancer regulons, pairs of attack features that become open and RNAs that get expressed down the line that regulate each cluster? And this is integrating known information about transcription motifs that are upstream of a given RNA. And what we were able to find very reliably is that TIM were much more enriched for AP1 factors, sort of consistent with what we found from our single cell atlas earlier. And what these enhancer regulons allow us to do, now that we have this information about pairs of attack features and RNAs that are co-regulating each other as a unit, we can now begin to model the entire gene regulatory network of a cell at single cell resolution in our entire data set. And once we have these cell-specific GRN models, we can actually do something very interesting, which is to perturb these cell models in silico. We can do transcription factor knockout and ask, okay, what if we remove this transcription factor from the system? What does this cell become? Does it remain in its current state, or does it revert back to a homeostatic state, for instance? And so we did this for a couple of transcription factors that we found particularly interesting. You can see FOS and NFKB2 up here at the top, for instance. These were nominated as some of the strongest transcription factors in E-regulons that were driving the TIM state. And we reliably found that when we knock these out in silico, they're predicted to revert back to a more homeostatic state, whereas it has no effect on DAM, which you can see here to the right, for instance. SOX5, which was a transcription factor that was predicted to drive DAM emergence, if you knock that out in silico, they also revert back, no effect on TIM. But what we found very interesting is that another transcription factor, KLF4, that was predicted to be relevant for TIM emergence, when we knock it out, there's no really cohesive effect, right? There are some changes, but you don't see a consistent return back to homeostatic state like we saw for FOS and NFKB2. So what this begins to tell us as a prediction is that while FOS and NFKB2 are required for maintenance of the TIM state, KLF4 might be required for emergence, but be dispensable for maintenance. Once you're a TIM, you no longer need to maintain expression of KLF4, but you might need to express it to become a TIM in the first place. And these kinds of predictions can be tested down the line, which we think is very exciting. But the natural next question was, we know that there have been a lot of single cell studies of microglia and different populations in Alzheimer's disease. And very often they find these populations of microglia or of immune cells that are present in murine models, but don't really exist in human models. And so DAM are a really good example of this. In every single murine model of Alzheimer's that you look, you see this massive inflation of this disease-associated microglia. They have a very clear canonical transcriptional program. Everybody knows what a DAM looks like. They're very easy to label in single cell. But really beautiful work from about 3 years ago from Stevens's group showed that if you look in human single cell data, there's really no equivalent DAM population. Instead, the signature is kind of distributed and spread across 5 or 6 different microglial states. And so our concern coming into this was, well, do TIM have a similar sort of analog in human populations, or is this going to be a DAM story all over again? And so to answer this question, we looked across 10 different single nucleus RNA sequencing data sets that have been previously published. So big thanks to the prior literature for making all this data available, doing a lot of the legwork here. And we did label transfer by anchor integrating our data with these other data sets and just transferring over the labels over to the nearest cell. And what we found, which was very relieving, is that every single one of these data sets had TIM at about 5 to 8% frequency, almost exactly identical to what we found, the 7% at the 1-year time point, the 60-week time point that we had for our multiome data. And what we found to be really reassuring is that if we looked at one of these data sets in particular, this is from the ROSMAP DLPFC2, this is from the dorsolateral prefrontal cortex of 465 human donors. So it's a really rich data set. And in addition to having all this single cell data, it's also labeled with the individual that it came from, which comes with a really rich set of metadata. So we have information about the individual's their sex, their disease severity, their age at death, things like that. But we also have information about their APOE genotype. So what this means is that we can now ask, what are the best predictors of TIM frequency on an individual level across these 465 human donors? What we find is that by far the best predictor of TIM frequency is E4 allele count. The more E4 alleles an individual bears, the more TIM they had. Exactly the same as we found in our murine data. What was really interesting is that we also found male sex was a strong predictor of TIM frequency, sort of unexpectedly, because we hadn't looked at gender as a question earlier. But of course, we know that gender is a big risk factor for Alzheimer's. Females get Alzheimer's disease much more frequently than males do, partially because they also live to be much older than males on average. But there's a lot of factors there. And then BRAC score, which is a proxy for disease severity. I'll note that age at death was not predictive here, but that's because almost all of these individuals were donors over the age of 85. So it's just not a particularly informative parameter in this particular analysis. But we have other data showing that age is still a predictor of a TIM frequency in human data. But to really dive deep and drill down into this question of whether TIM exists in human Alzheimer's tissue, we performed single molecule spatial transcriptomics using the Xenium platform from 10x to identify whether we could find TIM in their spatial context natively in Alzheimer's donor tissue from humans without having to dissociate and do single cell sequencing. And so we did this on 6 different human donors, 3 homozygous for E3, 3 homozygous for E4. And we were very relieved to find that we saw TIM, which are marked in these larger purple dots, very abundantly present throughout all 6 of our human donor samples. And we were also able to, because Xenium is non-destructive, after performing Xenium, we stained for methoxyxo4, which is a marker for amyloid beta plaques in the brain. And we showed that these TIM frequently colocalized with regions of high plaque density, which was very interesting. But what really excited us is that if we looked between E3 and E4, not only did we find more of these TIM in APOE4, we also found that these APOE4 TIM were much more frequently associated with cortical neurons. They were in the actual cortex, surrounded by neurons or surrounded by these astrocytes that express inflammatory markers, or next to other TIM. Whereas in E3, we instead found them much closer to the oligodendrocytes, further away from the cortical layers. So we begin to have this model that in E4 donors or E4 individuals, they have more TIM. Their TIM are more likely to be this effector low state, and they're more likely to be in the cortex surrounded by neurons and where the amyloid beta plaques are. Whereas in E3, they have fewer of these TIMs, and they're further away from the cortex, less likely to cause damage. But we now really wanted to get at functional questions. And the first of these that we did was to ask, well, how do TIM regulate the environment around them with respect to amyloid beta pathology? And so to get at this question, we harvested immune populations from the brains of AD APOE mice at 60 weeks of age, generated single-cell suspensions from their immune cells, and pulsed them for 15 minutes of fluorescently labeled amyloid beta. And the rationale here is that 15 minutes is enough for them to phagocytose the amyloid beta, but not enough for them to degrade it. And we can then do flow cytometry to split all these microglia into a low uptake and a high uptake pool, and then perform single cell sequencing, splitting everything up. So we have 6 different populations, E2, E3, E4, low uptake, high uptake. And we can ask, how does the interaction between your microglial state and your microglial genotype impact your ability to clear amyloid beta ex vivo? And we found, there's a lot more data here that I'm not showing you, but we found that TIM were reliably enriched in the low uptake pool, which is to say that TIM have a deficiency in their ability to clear amyloid beta from the environment. So we sort of implicate it in amyloid pathology very directly here. And the last bit of data that I'll share with you is that we did this experiment where we took these same mice, E2, E3, E4 Alzheimer's mice, aged to 60 weeks, and treated them with aducanumab, which you might know is a recently approved, FDA-approved therapeutic for Alzheimer's disease. It's an anti-amyloid beta antibody therapeutic. And so again, we have 6 samples here, E2, E3, E4, treated with control or with aducanumab. And we found, just like Wei showed before, that you have this massive influx of adaptive immune populations like T and B cells that come in from the meninges. But what we found that was most interesting is the effect that we saw on TIM. So whereas effector low TIM, which you can see in this area, really had no impact. There was no change upon aducanumab therapy. Effector high TIM were massively rejuvenated by aducanumab therapy. We looked at cell-cell communication in the system. The biggest targets, the biggest receivers of cell-cell signaling were indeed these adaptive immune populations, but the bulk of these signals actually came from these effector high TIM. So it appears that aducanumab therapy was able to rejuvenate this population and allow it to orchestrate the immune response to amyloid beta under aducanumab therapy. So what's the general story? Broadly speaking, we use single cell techniques to look at heterogeneity in microglia, quote unquote, beyond the M1, M2 paradigm, which of course is just a loose vision anyway. It's of course not a strict thing. It's more of a spectrum. But our view here is that we've always had this sort of homeostatic inflammatory paradigm. Now we're introducing a second dimension. You can't just go from homeostatic to inflammatory, you can also become exhausted. And this exhausted population is a terminal inflammatory population that enriches with Alzheimer's, with age, and with APOE4. And as I said, they look like an exhausted population. They don't do glycolysis as much. They have this weird combination of pro and anti-inflammatory signals. They even express SERPINE1, which is a canonical marker of senescence. And the long-term concept is that just as immune checkpoint blockade unshackled exhausted T cells in the tumor microenvironment, a similar idea might apply to microglia in neurodegenerative conditions like AD. There are 4 big questions that we're looking to answer in the future. The first is, how do TIM contribute to Alzheimer's pathology, if at all? Are they active participants? Do they release signals that modify or modulate the environment? Or are they passive? As you get more TIM, you just have fewer functional microglia. The second is, what biases E2 versus E4 to have effector low versus effector high TIM states? Number 3, whether this AP1 signaling that we identify is actually necessary and or sufficient for TIM emergence or maintenance. And if this is necessary, then this might be a druggable target to clear TIM from the brain and restore homeostatic signaling. And finally, this is the question that I'm most excited by. Do TIM exist in non-AD neuroinflammatory conditions? You might imagine that other diseases like Parkinson's or multiple sclerosis or brain tumors, in which you also have chronic inflammation in the brain, might also give rise to this exhausted microglial state. And so that's the manuscript. It came out in Immunity just over 2 weeks ago. If you have any more questions about some of the data we presented, I'd encourage you to read that. And I'll just end with my acknowledgements thanking the lab, Sohail in particular, who's an amazing mentor, Jose Leto, who now has a lab at the Medical University of South Carolina and was an amazing co-author and collaborator on this, the core facilities that help with the work, and my funding. And I'm happy to take any questions.